Oxidative protein folding in eukaryotes, consequences and mechanisms. Boppudi Vinoothna Ieshita Pan Department of Biomedical Engineering Assistant Professor, Department of Biotechnology Savitha School of Engineering Savitha School of Engineering. Abstract. The endoplasmic reticulum, ER, gives a domain that is exceptionally streamlined for oxidative protein collapsing. As opposed to depending on little particle oxidants like glutathione, it is presently evident that disulfide arrangement is driven by a protein transfer including ER1, a novel moderated fad-dependent catalyst, and protein disulfide isomerase, PDI. ER1 is oxidized by subatomic oxygen and thusly goes about as a particular oxidant of PDI, which at that point legitimately oxidizes disulfide bonds in collapsing proteins. While giving a hearty main impetus for disulfide arrangement, the utilization of atomic oxygen as the terminal electron acceptor can lead to oxidative worry through the creation of reactive oxygen species and oxidized glutathione. How ER1P recognizes the wide range of PDI-related proteins and how the cell limits the impacts of oxidative harm from ER1 stay significant open inquiries. Keywords: PDI, oxidative folding, secretory protein, ROS. Introduction. For the folding and stability of secreted proteins, the formation of intramolecular disulfide bonds is critical. This process involves thiol oxidation for disulfide bonds formation followed by rearrangement of non-native disulfide bonds. These bonds are regularly essential for the strength of a protein structure, and the impairing of cysteine deposits can forestall proteins from accomplishing their local adaptation, leading to misfolding. Reported in vivo process of protein folding which occurs rapidly. However, in vitro oxidative protein folding occurs slowly but in a spontaneous way, taking from hours to days. Exemplary tests by Anfinson et al. 1961, given proof that disulfide development is an unconstrained process and that the polypeptide itself is adequate for accomplishing the local state in vitro. In contrast, different parts of protein collapsing, disulfide connected collapsing is delayed maybe because of its reliance on a redox response, which requires an electron acceptor. These contemplations indicated that disulfide connected collapsing is a helped procedure in vivo, which was exhibited by the disclosure of DSBA freaks in Escherichia coli that showed traded off disulfide arrangement, Bartwell et al. 1991. 1. In eukaryotes, oxidative protein collapsing happens in the ER. Studies utilizing the great substrate ribonuclease A prompted the identification of protein disulfide isomerase, PDI, a protein that can rearrange incorrect disulfides as well as catalyze disulfide formation and reduction in vitro, Goldberger et al. 1963. Despite the ability of PDI to enhance the rate of formation, reaction remained unknown. Over the past 40 years, a number of different factors have been proposed to contribute for maintaining the oxidized environment of the ER, which includes the preferential secretion of reduced thiols and uptake of oxidized thiols, as well as a variety of redox enzymes and small molecule oxidants. Ziegler and Poulsen, 1977, Wang et al., 1992, Carl et al., 1997, Frand et al., 2000. However, the physiological relevance of these to oxidative folding has been unclear due to a lack of genetic evidence. 9. A combination of genetic and biochemical studies using the yeast Saccharomyces cerevisiae, and more recently mammalian and plant systems, have begun to reveal the proteins and mechanisms behind this fundamental protein folding process. Protein disulfide isomerase, PDI, which is an abundant catalyst for native disulfide bond formation in the ER lumen, contains 4 domains homologous to thioredoxin, 2 of which possess a redox active CGHC motif, Ferrari and Soling 1999. Depending on the nature of the substrate protein and the redox conditions of the assay in vitro, the active site cysteines of PDI can participate in dithiol disulfide exchange reactions catalyzing dithiol oxidation, disulfide reduction, or disulfide isomerization, reviewed by Friedman et al. 1994. In addition to PDI, many other yeast ER proteins like MPD1P, MPD2P, EUG1P, and the product of ORF-YIL005W exhibit the hallmarks of a thioredoxin fold, Frand and Kaiser 1999. 2. Apart from PDI ER1, a gene encoding a novel but conserved ER membrane protein required for the net formation of protein disulfide bonds, Frand and Kaiser 1999. It was already reported in a conditional ER1-1 mutant, secretory proteins remain in a reduced state in the ER but would normally contain intramolecular disulfide bonds, such as CPY, Ref. Evidence suggested that ER1P activity introduces oxidizing equivalents into the ER lumen. The thiol oxidant diamide can restore CPY folding and viability in ER1 mutants, and that overexpression of ER1 confers resistance to toxic levels of the reductant DTT. The conserved, ER-resident protein ER1P plays an analogous role to the bacterial periplasmic protein DSBB in oxidative folding. Both ER1P and DSBB specifically oxidize a thioredoxin-like protein, PDI in eukaryotes, DSBA in bacteria, that serves as an intermediary in the transfer of oxidizing equivalents to folding proteins, Bartwell et al., 1993, Frand and Kaiser, 1999, 2 et al., 2000. Molecular oxygen can serve as the terminal electron acceptor for disulfide formation in both prokaryotes and eukaryotes, Bader et al. 1999, 2 and Weissmann, 2002. Under anaerobic conditions, the DSBB-DSBA system can support disulfide formation via alternate electron acceptors, such as fumarate, Bader et al., 1999. However, in bacteria, oxidative folding is conveniently coupled to molecular oxygen through the respiratory chain. ER1P uses a flavin-dependent reaction to pass electrons directly to molecular oxygen, 2 and Weissmann, 2002. As a result, ER1P activity may generate reactive oxygen species, ROS, that could contribute an additional source of cellular oxidative stress, suggesting that its activity must be regulated according to the folding load. Furthermore, the evident particularity of ER1P for PDI may permit the various homologs of PDI to stay in the decreased state so as to complete separate redox works beside protein oxidation. This review will focus on the ER1P catalyzed mechanism of oxidative folding and its broader cell biological amplifications. 3. 2 oxidative folding machinery components in eukaryotes. Though the exact mechanism of oxidative folding is yet to understand and it is dependent on electron acceptor. Genetic screening in yeast revealed that membrane-associated ER protein ER1P, ER oxidoreductin 1, is a crucial component for oxidative folding, Frand and Kaiser, 1998, Pollard et al., 1998. Mutation in this protein causes huge accumulation of misfolded protein inside the lumen. They are found sensitive to DTT, Frand and Kaiser, 1998, Pollard et al., 1998, in vivo. Membrane association of ER1P may allow the protein to be retained in the ER and facilitate CO-translational disulfide formation. ER1P possesses 7 conserved cysteine residues that are likely involved in catalyzing electron transfer, Frand and Kaiser, 1998, 2000, Pollard et al., 1998. However, ER01P has no homology to any redox enzymes or other known proteins in human. There are two ER01 isoforms, HER01L also, HER01L, Kabibo ETAL, 2000, Pagani ETAL, 2000, which do not have a COOH terminal tail of 127 amino acids required by the yeast protein for membrane association, Pagani ETAL, 2001. In vivo, membrane acquisition of ER01P may permit the protein to reside within the ER to facilitate CO-translational disulfide bond formation. ER01P has 7 saved cysteine deposits that are likely engaged with catalyzing electron transfer. Friend and Kaiser, 1998, 2000, Pollard ETAL, 1BUT ER01P has no homology to any redox chemicals or other known proteins. Along with its major function of protein folding in ER yeast, ER01P and HER1LSS are induced by unfolded protein response, UPR. Friend what's more, Kaiser, 1998, Pollard ETAL, 1998, Pagani ETAL, 2000. The expression of HER1 law is controlled by hypoxia, Jess ETAL, 2003, however it won't control HER1LSS, 6. PDI has been known to help for the development of disulfide bonds. It has been appeared to catalyze disulfide bond arrangement also, isomerization, for a wide range of substrates in vitro, Friedman, 1989, yet its job in vivo still unclear. It is found that PDI is a fundamental protein which comprises 2% of the protein in the ER. It contains 2-thiodoxin-like cis-GLY his cis, CGHC, active site, Goldberger ETAL, 1963, Lavoisier ETAL, 1995, which is important for DTT sensitivity, Holst ETAL, 1997. PDI can't work as an oxidant of proteins yet can in any case catalyze the isomerization of protein disulfides if this active site is mutated. Moreover, in ER01-1 mutants, low accumulation of PDI proposed that ER01P worked upstream of PDI in a pathway for disulfide development in the ER, Friend and Kaiser, 1999. 10. The sulfhydryl oxidase ERV2P has been ensnared as a promising role in oxidative folding in the ER along with ER01P, Gerber ETAL, 2001, Severe ETAL, 2001, Net ETAL, 2002. ERV2P is an individual from the F/ALR group of sulfhydryl oxidases, Thorpe ETAL, 2002, which have been found in various subcellular compartments. Despite the fact that ERV2P can make up for surrenders in ER01P when overexpressed, Severe ETAL, 2001, and can oxidize proteins legitimately in vitro, Gerber ETAL, 2001, under the conditions analyzed, cancellation of ERV2 at most modestly affects development, Severe ETAL, 2001, 2 and Weissmann, 2002. The sulfhydryl oxidase action of ERV1P might be significant for appropriate mitochondrial homeostasis also, the get-together of iron-sulfur group proteins, Lasowski, 1994, Lee ETAL, 2000, Lang ETAL, 2001, maybe by catalyzing disulfide bond development, despite the fact that the oxidative condition of the mitochondrial intermembrane space isn't all around described. 7. These perceptions build up that ER01P is the major player in protein oxidation and that the physiological job of ERV2P is. A sulfhydryl oxidase is restricted to a subset of unnecessary proteins or development conditions not yet inspected. In yeast, there is a fundamental homologue of ERV2P, ERV1P, that is limited to the mitochondrial intermembrane space, Lang ETAL, 2001. Strangely, a vaccinia infection protein of this family has been involved in the arrangement of disulfides and viral proteins during gathering in the half-cytosol, Senkevich ETAL, 2000. 4. 3. The ARO1-dependent pathway for the transfer of oxidizing equivalents. Disulfide arrangement mediated by ER01P with PDI at sub-stoichiometric ratio proposed faster folding evidence with PDI, 2 ETAL, 2000. ER01P catalyzed oxidative folding continues quicker than the PDI catalyzed oxidative collapsing of substrates within the site of an ideal glutathione redox cushion, 2 ETAL, 2000. Moreover, this ARO1P-driven response can happen autonomously of glutathione both in vivo and in vitro, Quatso and Kaiser, 1999, 2 ETAL, 2000. A mutant PDI with cis-GLY Hizala, CGHA, active site is found a prevailing inhibitor of this ARO1 catalyzed response, thereby producing a mixed disulfide cross-connect both in vivo and in vitro, Friend and Kaiser, 1999, 2 ETAL, 2000. These perceptions show that ER01P oxidizes PDI legitimately through disulfide trade, 2 ETAL, 2000. PDI in this way catalyzes the development of disulfides and folding proteins. ER01P doesn't effectively go about as an immediate oxidant of folding substrates and in this way depends on PDI to move oxidizing reciprocals, 2 ETAL, 2000. Thus, by means of the protein transfer, ER01P to PDI to protein the exchange of oxidizing reciprocals happens. ER01P may likewise be engaged with intervening the retrotranslocation of proteins to the cytosol by oxidizing PDI, changing its compliance, and in this manner adjusting its liking for specific substrates, Psi and Rappaport, 2002. 5. For the role of glutathione in oxidative folding, glutathione is the major redox buffer in eukaryotic cells, Wang ETAL, 1992. The ratio of reduced, GSH, to oxidized, GSSG, glutathione is 100:1 in the cytosol, Wang ETAL, 1992. This highly reducing environment disfavors disulfide bond formation. However, in the ER where disulfide formation occurs, the ratio of GSH-GSSG is much more oxidizing at 3:1, Wang ETAL, 1992. This abundance of GSSG in the secretory pathway was long thought to be the source of oxidizing equivalents for disulfide formation, Wang ETAL, 1992. However, ARO1P catalyzed disulfide formation proceeds independently of glutathione both in vivo and in vitro, Quatso and Kaiser, 1999, 2 ETAL 2000. What then is the role of glutathione? Genetic evidence in yeast has demonstrated that glutathione is dispensable for disulfide formation and instead capacities as a net reductant in the ER, Quatso and Kaiser, 1999. In a screen for silencers of the temperature affectability of the ER01-1 freak, an erasure of GSH1, which is included in the biosynthesis of glutathione, was found to firmly stifle the ER01-1 phenotype, Quatso and Kaiser, 1999. The translation of this perception is that non-appearance of glutathione as a reductant brings about less decrease of oxidized PDI in proteins, which permits an undermined ER01 oxidation framework to help development. In GSH1 strains, the oxidative collapsing of CPY continues with typical energy, however is profoundly delicate to oxidative anxieties, steady with the job of glutathione as a net reductant, Quatso and Kaiser, 1999. What is the reason for the high GSSG content in the ER? ER01P can't legitimately oxidize GSH to GSSG, 2-ETAL, 2000. Be that as it may, ER01P and PDI can drive the oxidation of collapsing substrates even within the site of decreased glutathione, GSH. After some time, a progressive creation of GSSG coming about because of GSH intervened decrease of disulfides in PDI and collapsing proteins is seen in vitro, 2-ETAL, 2000, Fig 1. In this way, the wealth of GSSG in the ER is likely a result of ER01P action, and the GSH-GSSG redox cushion in the ER speaks to a balance between the results of ER01P intervened oxidative and glutathione intervened reductive procedures. The dynamic transporting of oxidizing reciprocals by ER01P and PDI that happens autonomously of the mass redox condition could clarify how the ER bolsters quick disulfide arrangement while keeping up the capacity to diminish or revise erroneous disulfides, maybe through glutathione and certain PDI homologs. The role of fad in oxidative folding. The reliance of oxidative collapsing on fad characterizes a novel job for this flexible redox particle in the ER lumen. ER01P is the primary depicted flavoprotein restricted altogether inside the ER lumen, and the presence of a hardy vehicle framework that brings fad into the ER lumen has been recommended. In yeast, oxidative collapsing in vivo is profoundly delicate to explicitly free cell fad levels, 2 in Weissmann, 2002. This affectability could be reiterated in vitro, as the movement of ER01P shifted fundamentally with little deviations from physiological fad focuses, 2 in Weissmann, 2002. Despite the fact that this affectability of ER01P to free fad levels is abnormal for a flavoprotein, it might give a way to control oxidative collapsing, see additionally beneath. Further work will be required to decide how fad regulates the movement of ER01P and regardless of whether this is a preserved property of ER01P. 8. 6. Conclusion. Oxidoreductases in the ER are responsible for net oxidation of protein cysteines with proper connectivity. To achieve correct cysteine pairing, disulfide isomerization or cycles of reduction and reoxidation may be necessary. Furthermore, to maintain the homeostasis, proteins oxidized and transported through the secretory pathway in the ER performs reduction and retrotranslocation of misfolded proteins. A major outstanding question related to biosynthetic disulfide bond formation in the ER is how the physiological functions of oxidation, rearrangement, and reduction of disulfide bonds are distributed among PDI family proteins. In conclusion, while past investigations of oxidative folding have concentrated on the mass redox capability of the ER, it is currently apparent that it's a protein relay. By the kinetic shuttling of oxidizing equivalents to folding substrates, eukaryotic disulfide formation proceeds as the ER01P-driven oxidation apparatus is protected from the mass redox condition. Decreased glutathione, maybe certain PDI homologs can aid the isomerization and reduction of incorrect disulfide bonds formation. Oxidative folding is coupled to the solid decrease potential of atomic oxygen through a fad subordinate system, but a potential result is the creation of harmful receptive oxygen species. Undoubtedly, ER01P could be a noteworthy supporter of cell oxidative pressure, and recommends that its action must be balanced agreeing to the folding load on the ER. Controlling the level of free ER01P might be a way to control oxidative folding as indicated by the cell's nourishing or metabolic state. Future work will uncover how the ER keeps up its ideal condition for the large number of redox forms required for the best possible collapsing of secretory proteins. 11.